So I'm going to talk to you about cellular communication. Before we get into it too much, I just want to make sure that everybody understands that pretty much all of the cells within a human contain the entire complement of genetic instructions. So they have the library really to be able to make any protein that any other cell within the organism has the capability of making. Okay, and so what this means is that a liver, even though it's really specialized, the cells within that liver have instructions to do some of the things that a heart cell could do. But these cells, when they're in their adult state, are differentiated. They've been instructed to behave in a very specialized way, and in that way they're only reading just a portion really of the instructions that it takes to create an entire organism or a human being. So when we look at cellular communication is really important for a couple different processes. Number one, you'd like to be able to communicate to neighboring cells, if conditions are changing. For bacteria, it's incredibly important to be able to get an idea about how many of the neighboring cells around you are bacteria. We call that quorum sensing, and that's just one example of how these cells are talking to each other. Also, we would like to be able to control the cell's ability to, actively divide or not divide, or turn on certain protein synthesis operations, shut down other operations. This kind of communication is done entirely with chemicals. Okay, and we'll show you a couple different examples here. Let's get into it. So with autocrine, what you have is the cell is going to signal itself. So in this instance, we have our ribosomes here studying the endoplasmic reticulum, and in this eukaryote, what's happening is there are proteins that are being produced. They're put into vesicles. The vesicles are fusing with the membrane, and the protein in this case is being released to the extracellular space. Now, if that protein reaches high enough concentration, it's going to be able to attach to a receptor on this cell's surface. And that might indicate to the cell that there's enough of this chemical in the extracellular environment and shut down the protein synthesis operation. If enough of this protein is absent, then the operation will get turned back on. And you can see how that might be beneficial for a cell kind of understanding or controlling the amount of protein that's produced within its local environment. That kind of signaling, again, is called autocrine because the cell is signaling to itself. Okay. A second type of local signaling is going to be paracrine, and this is when a cell signals an adjacent cell, a cell next to it. Okay, so for this example, I want to go through and use a motor neuron. So that is involving two cells that are very specific. It's a very highly specialized interaction. So the neuron itself has dendrites out here. This is a cell body, and it's going to receive the information and send it down this long tube, which is called the axon. Now, down at the end of the axon, we have these synaptic bulbs where a neurotransmitter is going to be manufactured, produced. Again, it's going to be put into vesicles, and the vesicles fuse with the membrane in the synapse and excrete the neurotransmitter out into the environment. Now, in this case, the neuron is not going to ever touch the adjacent cell. There's going to be a gap there. So it's going to release this chemical, this protein in this case, acetylcholine, across the synapse, and that's going to attach to a cell surface receptor, and because it does, it is received by this muscle tissue. What's going to happen is the muscle is going to contract. Okay, and so this is local signaling. The cell is not signaling itself, it's signaling an adjacent cell, so we call it paracrine signaling. Okay, a very important concept to get is that not all communication is happening over short distances. Most of the communication that's happening is actually traveling over large distances. Your brain is controlling a lot of the activity inside your body, and we have hormones that are going to travel large distances throughout the body here. And so this long distance signal is called endocrine signaling. Okay, so this is just a point of reference here is the pancreas. All right, and I'm just trying to show or depict like the flow of blood up into the arms and down into the legs. Okay, but in terms of the endocrine system, I wanted to go over the major glands just so that we feel comfortable with where they're located and pretty much what they're doing here. So this area of the brain, which would be centrally located right about in here, okay, this area of the brain is called the HPA axis. hypothalamus pituitary axis, and that really is where all endocrine function is controlled. The pituitary gland is considered to be the master gland of the endocrine system, and it actually secretes tropic hormones that are going to have impact or be able to change what is being secreted by all the rest of the accessory endocrine glands that are found dispersed throughout the body. Okay, so going from the top of the body where we have this HPA axis down, we'll just go through some of the major glands of the endocrine system. This is called the thyroid gland. It's located right here in the throat, and inside the thyroid gland you have the parathyroid. Those two things again are located right there in the neck area, and they produce some incredible hormones that are utilized throughout the body. Thyroxine, parathyroid hormone, to name a couple. Okay, as we move down, right about in this area, just above the heart, we have the thymus. Okay, moving down a little bit further, right in here, we have the pancreas, insulin, and glucagon, two most important ones to make sure you're able to recognize there. As we move down further, right about here in the back of this human's body, on the back wall, you have two kidneys, and on top of those kidneys reside the adrenal glands. Adrenaline is probably the most famous, but also cortisol is secreted by these adrenal glands, very important stress hormone. And then lower down, if we have a male, then the testes would be a very important endocrine gland for the development of male features. Also control of local testosterone levels. In females, obviously the gonads would be the ovaries, and they'd be a little bit higher up into the body. So I hope that makes sense to everybody where these things are located.
And so we need to have a mechanism for long distance travel. So instead of inventing its own, the endocrine system utilizes the bloodstream. And so endocrine glands are going to secrete chemical hormones, either peptide, protein hormones, or steroid, which are lipid hormones. They're going to produce those, they're going to excrete those into the bloodstream, and then they're going to be able to circulate in the blood.
Okay, let's get into how cells are able to send and receive signals. Okay, so in order for a signal to be received, what has to happen is a chemical has to be released. We just went through a couple different examples of this, but the chemical that is released into the bloodstream or into the local environment, we consider that chemical to be a ligand. The ligand can be either a protein or it can be a lipid. So the ligand, once it's secreted, is then going to react with a very specialized receptor. So if it's a lipid ligand, a steroid like estrogen or testosterone, it's actually going to go directly through the cell membrane, and it's going to react with an intracellular receptor. Okay, and so you can see here that the shape of this intracellular receptor is specific to this steroid. Okay, I want everybody to kind of get that if the shape of this was off, the steroid would not be able to change or to bind with, react with this intracellular receptor. Okay, so it's specific in that nature. Okay, if it was a protein, that protein is going to be able to bind with this specific aspect of this surface receptor. So protein receptors are only going to be found on the cell surface. Okay, they're extracellular receptors, whereas steroid receptors are going to be intracellular. Again, both have to be highly specific so that not every molecule that comes by is going to interact and cause a change in that receptor molecule. Okay, and this part of cell signaling we call reception. So at this point, the signal has been received by the cell in either case. Okay, now this is a very specific type of cell reception. I want to take you through the process here. It's actually part of what's known as a signal transduction cascade. Now, a couple of things about this I just want to point out. You have a multi-step pathway with a signal transduction cascade, and this is important because it allows it for amplification, it allows for finite control. It also is going to allow this ligand to attach to the surface receptor of this cell and have one reaction, and then if this was, let's say, in the liver, that same ligand can bind here, but what happens inside the cell can vary quite a bit, as opposed to maybe the same ligand binding on the surface of a muscle. What happens inside can be very different. Okay, so hopefully that will make more sense or resonate more as we go through this. So the ligand is going to attach. This is called a G protein complex because we have a guanosine diphosphate here attached as part of this complex, and so the ligand is going to attach here and cause a conformation change within this protein. That means the protein is going to change shape. When that occurs, it's going to kick out the guanine diphosphate and allow for the guanosine triphosphate to be attached here. So this is important because the guanosine triphosphate is the activated state of this G protein complex. This thing now is on, it's active, and it's going to be able to take this initial signal and move that signal into the cell. Okay, so the guanosine triphosphate is going to break free and going to move elsewhere into the cell. Okay, so we have guanosine triphosphate here. It is charged, it's ready to go. It's very similar to ATP in that we have 3 phosphates. Okay, and so it's going to be capable of phosphorylating other proteins inside the cell, carrying that message further on. Okay, so in this case, it's going to join up with an adenyl cyclase, and this is like a factory inside the cell. When GTP attaches to it, it turns this factory on. It's able to fast a phosphate group to this molecule, and when that happens, that phosphorylation allows for ATPs to be taken in, and a very important, a critical secondary messenger to be released. This is called cyclic AMP. It's one of the most important secondary messengers to be able to recognize and understand. And for right now, I need you to understand that the signal has moved from the outside of the cell now to an operational function within the cell. This cyclic AMP is going to be able to be utilized to do work elsewhere in the cell. Okay, and so cyclic AMP will go on to turn on certain protein production functions, or to shut down certain activities within the cell. And that's the part that's going to allow us to vary from cell to cell, what the end result is, what is the response by the cell. But the guanosine diphosphate, once enough cyclic AMP has been produced, is going to go back and reset this G protein complex. So now it's going to be able to receive another signal from a ligand, turn this factory back on if need be. Okay, so that's pretty much a signal transduction cascade. There's a couple things that I just want to make sure that we feel comfortable with at this stage. Okay, first off, a cascade is going to involve a multi-step pathway. Okay, so it's not just one step. In this case, I showed you the G protein initiating the signal reception, taking the guanosine triphosphate and moving that information inward to trigger an adenyl cyclase. But in other cells, you can have 15, 16 steps of a pathway before response is initiated within the cell. And this is really important because each of those steps allows for very finite control of the response. It also allows you to amplify the signal so that more of a cellular response can be had. And I also just want to point out that in any of these pathways, we can have 2 molecules that play crucial roles here. One of them is called a kinase. A kinase, if you recall from previous conversations, is going to be an enzyme responsible for phosphorylation. So what that means is it's going to be adding a phosphate group to a protein. Now that phosphorylation is going to change the shape of the protein and therefore its function, right? And we also have phosphatase molecules that are responsible for removing phosphate groups from molecules. So those 2 things are highly involved in most signal transduction cascades, and you'll see a series of kinases, protein kinases, that will carry the initial signal from the point where it's received to the response. And I would like everybody to also feel comfortable with the fact that if any of these proteins, you can envision maybe the receptor here, if this protein was altered in terms of its structure, its conformation, that ligand is not going to be able to bind there. And what do you think would happen if the ligand is not able to initiate the signal? Is the G protein going to get activated? None of these things are going to happen. Okay, so if we have any of these proteins get altered, then it can really alter what is the end result in terms of response to that initial signal. And that's any protein in the pathway. So if one of the kinases is now inactive because of change in conformation, the signal, even though it might be received up here, is not going to make it to its end response. I hope that's clear. Please ask questions if not.